Hello students, welcome to Learner's Planet. Today, let us again start with the class of biology. And the topic that we are going to start today is microorganisms. Micro, which is actually a unit that you must have studied in your earlier classes, like centimeters, meters, millimeters or micrometers. So micrometer, it is actually a unit which is very small, even smaller than the millimeter. So you can imagine how much small it could be. And if something is so small, then you can imagine that how we are going to visualize them. So today we are actually studying such a small kind of organisms. What are organisms? That we have already studied in our previous classes. A living system which is capable of performing the life-sustaining functions, like they can reproduce, they can grow, and so on. Now these organisms who are so small in their size, they can be our friends as well as they can be our foe. Now what we mean by this friends and foe is that they can actually help us or they can cause some harm also. So such type of organisms, they can have different type of role in our daily life. So in general, we must have seen that we all are surrounded with living and non-living things. So what do you mean by living and non-living things? Non-living things which cannot grow, which cannot show the chemical reaction on its own, which cannot reproduce. So what are the living things which are actually showing such type of properties of their own metabolic reactions? They can grow, they can reproduce, and so on. There are many other characteristics. But what we are going to give stress in today's lecture is that these bodies which are present in our surrounding, they are affecting our life in some way or the other. Further, there are several bodies in our surrounding which are actually present but they could not be seen. And if such bodies are able to multiply themselves, then they have to be a living thing. So such living organisms which are actually present in our surrounding but they could not be seen with the naked eyes, they are known as microorganisms, or these could also be known as microbes. So these microorganisms, we can define them as those living organisms which are present in our surrounding but they could not be seen by the naked eyes. So now the imagination begins. We are saying the little bodies are present in our surrounding but we are not able to see them. Then how they must look like? What should be their appearance? If they can reproduce, what could be the mode of reproduction in them? Then what is the number of such organisms which are present here? How they are going to affect our life? Many questions will come in your mind now. We will begin one by one to deal with solving these all problems or the imaginations or the questions coming in your mind now. So the first thing is that how they should appear, how they must look like. For that, if we have to see these objects or the organisms, we have to magnify. Here we have shown a magnified image of the microorganisms. Now you can see these bodies. Some are cylindrical and some are showing the spikes over its surface. So from this we can quote that the organisms can have different kind of shape. Besides, their surface could be smooth or it could be rough. And when we are magnifying it, that means we are increasing the size in terms of its image. We cannot increase the size of organism directly. We can only increase or magnify its image so that we can observe them properly. We can study them thoroughly. So here is one such image just to explain that how they are going to appear if they are magnified. But all microorganisms do not have this kind of shape only. They have various type of shapes depending on their type. Now, the next thing is that we are saying they are so small in their size, and when they are so small, they could not be seen with the unaided eye, or they could not be seen with the naked eye. Then how we can see them? How we can magnify their image? That is the next thing now. And for this purpose, there is one instrument called as microscope. So this microscope is a device or a tool or the instrument which is used in the laboratories so as to magnify the image of these little microorganisms. What is this microscope? How is it functioning? Now that is the next question that comes in your mind. We are saying it is microscope. It is a device to magnify the image of the organism. Now just think at once, you must have visited some eye specialist, a doctor, ophthalmologist. Why we go there? Normally, you all are so studious, right? So, while studying, due to such type of activities or watching TV, our eyesight is actually affected. When we visit the doctor, eye specialist, what does he do? He checks our eyes and he is providing us with the glasses if your eyesight is not fine. It is a general example we are quoting here. It's not necessary we all have these type of problems, but if we have, then this is the solution. And if the doctor is suggesting that now you have to wear the glasses, now those glasses, they are actually fitted with the lenses. These lenses will help us showing the images which are not clearly seen at long distances or at short distances by changing the magnitude of the image. So these lens actually helps us in magnification of such type of objects. Now we can relate that with the microscope also. There are these things which are the living organisms which are present in our surrounding and we cannot observe them. So we need some lens to magnify those images, to the images of those organisms. But such type of lens is actually not provided us by the doctor. The doctor is never going to give you the special glasses which are fitted with the special lenses so as to observe such type of microorganisms. And for solving this particular problem, the device has been discovered. Which device? It is microscope. So, here is this picture which is showing the microscope. It is a diagram, it is a picture of compound microscope. There are various kinds of microscopes like simple dissecting microscope, compound microscope, electron microscope. But what we are explaining here is a compound microscope which is normally used in biology labs. In this case, you can see the structure. Here it is provided with this long tube which is holded by this particular handle, and this handle or the entire instrument is placed over the horseshoe shape like structure. Now this is the outer design, but now let us understand the functional design of the microscope and the different parts which are present here, and definitely how do they function. So let us start with the parts one by one. The very first one is eyepiece. This eyepiece is the uppermost or the topmost part which is first of all seen, observed in the microscope. What is this eyepiece? It is a kind of lens. It is a lens from where we are peeping into the window, we can see the image of the object. Object, that is the microorganism placed under it, or any other object that we have to observe here. A little small thing. Now eyepiece, what it is? A glass or lens from where we are observing the image of the microorganism. Next here is this body tube. This body tube is actually a cylindrical path which is actually providing the path for the light to travel. and from where the image is actually observed. So this is just a path from where the reflected rays are actually passing through, and that is the thing which is forming the image and which is observed from the eyepiece. The next here is objective lens. So what is this objective lens? Objective lens, we can say it is a magnifying glass. So these magnifying lenses are fitted under this particular body tube. You can see this round structure here. This round structure is actually the nose piece over which the objective lenses are fitted. And these objective lenses are having capacity of magnifying the image. So the magnification of image is actually done by the lenses here. So these three lenses we are highlighting here, these are actually the objective lens. In a compound microscope, generally it is provided with the lens with varied power, and that is having the magnification power for 10 times, 45 times and 100 times. What we mean by that is, this particular lens can magnify the image up to 10 times. The one which is 45x, the second lens, it can magnify the image up to 45 times. 100, similarly, it can magnify image up to 100 times. So these are actually the specific function of the objective lens. In a microscope, the most important magnification type of function is actually performed by the objective lens, and the observation is done from the eyepiece. Now, what we have to do is, we are saying we are magnifying the image of certain object. The question is where this object is, where we are, from which object we are actually magnifying, where it is placed. So that is another part which we are showing in this particular picture. So we can see this particular heading here, that is a slide with a specimen. So the specimen is the object, or it is a sample present in the biology lab. Or in general terms, we can say the things we are observing in the laboratory, they are termed as specimen in biological terms. So here we are saying that it's a slide with a specimen. So these specimens, or the microorganisms which has to be visualized, it has to be sampled over a surface which is provided by the slide. Slide is nothing but the rectangular glass structure. These small glass structures which are having the rectangular shape, you can see it is placed here. So this rectangle structure, this is actually used for placing up the microorganisms. So it could be placed over there. Next is from where this specimen is coming. We said that these microorganisms, they are present in our surroundings. So they can be collected from the surrounding only. So let us quote one example. If we have to observe the microorganism present in the water, so what we have to do? We have to collect the sample of the water which we want to observe, which we want to study. And from that, just a small drop of water or sample is placed over the slide. And then if staining is required, that means if it has to be treated with certain dyes, then the staining process is done, else it is covered with a cover slip, a round small glass structure. So that would be placed over this, and then it is going to be placed over the stage. This rectangular part present in the microscope is known as the stage, over which the slide is actually placed. Then we have to fix the position of the slide also, and that is done with the help of these clips. You can see these screws which are having attached certain clip-like structures. There are pair, and this pair of clip is going to hold the slide over the stage. Besides that, this stage is having a hole in the center, and that is definitely provided with the normal glass. Why this glass or hole is present in the stage? Because in the dark we cannot form the image of this particular object. For that, we need the light source. We need the light rays. So, underneath, under the stage, a mirror is present. You can see this mirror here. This mirror is going to reflect the light rays towards the stage. And from this particular hole, it will be transferred, it will be passed towards the specimen present over the slide. Now, just imagine from how this mirror is going to reflect the rays. For that, definitely we need some light source also. So, these light rays, they can either come from sun or they can be reflected or they can be coming from the another thing that we have to we must understand at this level is that when we are using these light rays, then intensity should be moderate. That means the amount of light which is reflected towards the slide, it should be in control. It should not be very dim. It should not be very bright. It has to be according to the tolerance of these organisms or the magnification. So, the amount of light, how much it has to be transferred towards upside, that is towards the slide, that is controlled by the another structure present below the stage only. You can just see the small round structure here. This particular kind is small, this one. So this particular structure is actually helping in controlling the intensity of light that has to be transferred towards the specimen. Besides that, there we said here is a handle and a horseshoe-shaped structure which is used for handling of this particular instrument. Besides that, over this handle the knobs are present. These knobs are a kind of screw which can actually move this body tube up and down. Why up and down? Because the distance between the lens and the slide has to be maintained. So this distance and the focus is actually maintained by such type of adjusting screws in the instrument. So this is how the microscope is actually functioning. And if such provisions are there, then definitely we can magnify the image of the organisms, the microorganisms.